Well, hello. My name is Brant Isakson. I'm a professor of molecular physiology and biological physics. I'm also a resident faculty of the Robert M. Byrne Cardiovascular Research Center here at the University of Virginia School of Medicine, and that's in Charlottesville, Virginia. I'm also the editor-in-chief of the Journal of Vascular Research. So my research is really focused on how cells in the small blood vessels talk to each other. So cell-cell communication. And usually we look at the small arteries, either the small veins or the capillaries, and what do they use to talk to other cells in the surrounding environment, whether that be molecules that are released from the cell, so more of a paracrine type communication, or the type of direct cell-cell contact, like things that are mediated by gap junctions. How do the cells use these proteins to talk to each other? This is really important because the ability of cells to constantly communicate with each other are a way for the tissue to act as one syncytium, how to control blood flow, how to control blood pressure. And so when we start disrupting the way that cells talk to each other, we can have lots of disease states start to appear. So that's really the crux of what my lab studies. So we have in our research, the primary focus is using mouse models. We also use cell culture, but we also do have a couple open IRBs, which are human research trials, very small. And usually these are for tissue, so we get some data from humans on their blood pressure, usually they're treatment-resistant hypertension individuals. And then we take biopsy samples from them and see how the blood vessels are different from these humans, or how in some of our more recent papers, we've extracted their RNA and their protein to compare and contrast how that looks to a human. We've also developed, I think maybe more importantly, we've patented a few molecules that we've been able to test on some of these human patients, and been able to kind of really start at the basement level, trying to see how some of our work could be translated. So we have a lot of those kind of ongoing small human trials in the lab. So I'm associated with quite a few different scientific organizations. Those include what's called NAVBO, North American Vascular Biology Organization. I've been involved with Microcirculation Society in the United States for a long time, also with the American Physiological Society, APS, and also with the American Heart Association. And also we've had some work with the European Microcirculation Society. So, over the years, I've had various roles in these different organizations. Mostly, I've served as counselor for the Microcirculation Society, and I've been chairing the programming committee at the American Physiological Society for the last 2 or 3 years now. So I'm an active member in all of these organizations, and so we go to the meetings, and we try to promote the best possible science. So I think a huge benefit for these organizations is not only the critical feedback you get from other scientists who know what you're doing, so they're in the field of microcirculation, or they're in any of these societies. They're going to be very close to the type of research that you do, so they're going to be able to give critical feedback on the work that you're doing. In a lot of ways, these are really great places for trainees to be involved. All of the organizations I've listed, MicroCirc, APS, NAVBO, AHA, they all have really, really great programs for trainees. And so not only financially to get them to travel to the meetings, but in terms of groups of, they try to start forming these contacts between trainees early, and mentorship type programs that a lot of them have, where you'll go have a coffee or a meal at the meeting with some of these mentors, and just start asking some questions about the best way to do research. How do you deal with certain problems that arise? So I listed multiple organizations, and they all have these excellent trainee programs that I'm very, very enthusiastic about with the trainees in my lab. So I would say that not only the science, but the ability of the trainees to interact with other trainees is especially important. So just this year, I was awarded the Robert Byrne Mentorship Award from the American Physiological Society. And that's something I'm especially proud of. And it has to do not only with the quality of your research, but the quality of your trainees. Really, that's the thing I'm the most proud of, is the trainees that have come through my lab. And so that was an award that was instigated by former trainees that were in my lab, who've all done exceptional work. And so that makes that award especially meaningful to me. Well, I do think that there's really a sense of, I don't know if this is not the right word to use, but a sense of family with the publisher. It's not one of these kind of corporate entities that are really looking for the bottom line all their time. They're just trying to do the best possible science to the academic community, and I really appreciate that. And so that's something that I really respect about the company, and something that I always talk about in terms of, we try to do the best possible science, and Karger just lets us do that. And I think that's really something unique, especially in this day and age, with the proliferation of journals. It really helps Karger stand out in terms of focusing on the best science, the most rigorous peer review. And so I think that would be something that I would add. What I'd like to do is expand the journal even further, and I think that that's something that we could definitely do in collaboration with Karger. And the other thing is to start making the journal open access. It's somewhere that we're moving. The open access journals is something that we're moving towards, and I think that that is something that will really benefit the journal and the scientific community in general. What I'd like to do is continue to promote the rigor of the science. I think that that's something that we can hold our heads high in terms of the journal, is that we really wanted to promote this rigorous peer review, this rigorous evaluation of the manuscripts, but also do it in a very timely way. So really the two things we talk about a lot, that we continue to work on, continue to expand, and I think we've done really well, but is to really push on the rigor of the manuscripts, but also get the turnaround of the journals, really the decisions, as quick as possible. I mean, because that's something that, being a researcher myself, is something that I really would like to know very quickly, is how well the journal fares, how well the manuscript fares in peer review. But I also want to make sure that it's been critiqued in a way that is fair, but really rigorous, and makes my paper that much better. So that's something that we will continue to do. And we are starting to expand our editorial board and expand our associate editors to really not only help with the rigor and the time for review, but also to make sure that we have a consistent peer review network that knows what we're looking for at JVR. So these things are something that I'm really pushing for. I'm really looking forward to continuing to expand with the journal.